Morning, everyone. I'm Jeremiah. I'm an MD PhD student in the lab of Neil Hayes in Memphis, Tennessee. And today I'm going to talk a little bit about, my project involving the integrative genomic analysis of one of the largest HPV-associated head and neck cancer cohorts. So just a bit of an outline for any of you who saw my talk on Wednesday. The background is going to be a little bit repetitive, but I promise if you bear with me when we get to the results, there will be some new information I'm going to share. Focusing mostly on the cohort demographics as well as how we came to a list of some of the most significantly altered genes in head and neck cancer, and then we look at the patterns of those genes as well as the molecular pathways associated with them. And then lastly, maybe talk a little bit about our future plans. You guys are all experts here, so I'm not going to focus on this, but what I want to mention is that HPV is the most important subset of head and neck cancers, but is largely unexplored in large-scale integrative genomic studies. This is some results from the Cancer Genome Atlas head and neck cancer study that was published in 2015. And while it was largely successful in identifying driver mutations in a majority of patients, you'll see that there are quite a few patients who lack driver alterations. And then if you look more closely, you'll see that most of these are human papillomavirus positive head and neck cancers. So what we're really trying to do is figure out, are there any driver alterations that we're missing due to large or lower sample sizes of human papillomavirus associated head and neck cancers. This is another figure from the TCGA study, and it shows some of the most commonly altered targets as well as the pathways that these are involved in. And what we've been able to do is add about 250 more HPV positive cases and try to expand on these pathways and add some new players, as well as try to nail down some of the details that was missing from the Cancer Genome Atlas. Largely in cell cycle control pathways involving the HPV proteins, as well as some immune regulating pathways involving interferon signaling and viral clearing. So a little bit about our technology here. We do targeted hybrid capture sequencing in FFPE tissues, and we are able to do really deep sequencing, so that allows us to detect many alterations even in some lower purity samples. However, our assay is relatively good at identifying most of the driver alterations, but there is a chance of false positives due to the nature of the preservation as well as any other artifacts that occur during sequencing. So there is some filtering that has to go on. But even past that, even if an alteration passes filtering, it's still important to classify that alteration as being important in cancer. And one way we do that is trying to identify driver events, which are most likely to impact cancer initiation and progression. And these tend to be either loss of function mutations, such as nonsense or frame shift deletions or insertions, as well as hotspot mutations that are in oncogenes. So, like I mentioned before, there are a proportion of samples that lack known driver alterations. So is this a question of sample purity, or is this really an opportunity for discovery? So our cohort here is a novel cohort, and about half of the patients are HPV positive, which is great. And you can see that a majority of these HPV positive patients are oropharyngeal, but there are a handful of other anatomical sites, such as the larynx and the oral cavity. So essentially, we started with a list of genes identified by the Cancer Genome Atlas, as well as other studies, and then we were able to add many targets to this list that are affected by recurrent copy number alterations, as well as somatic mutations. So using a synthesis of all these properties, we were able to get to a list of about 99 genes, which is quite large, and it's pretty hard to deal with that many genes at once. So one of the first strategies we tried to do was group them into pathways. One thing that grouping them into pathways lets us do is understand a little bit more about the biological implications of these alterations, but also allows us to group them and see patterns such as co-occurrence or mutual exclusivity. So the first pathway that I'd like to talk about is the cell cycle pathway. And you can see some pretty striking differences right away between the HPV positive cases on the right with the purple bar and the HPV negative cases. The HPV negative cases are affected by canonical loss of function mutations in P53, as well as deletions and loss of function mutations in CDK and 2A. Both of those are tumor suppressor genes. If you look on the right in the HPV cases, you'll actually see more targeting of the E2F signaling and driving the cell cycle through that pathway. So you can either amplify E2F1 directly, or you can lose some of the regulators of E2F1, such as RB1, which is a target of HPV protein E7, as well as RBL1 and 2, which are also targets of the HPV protein. So not only do we see mutations that are loss of function in black, but we do see quite a few deep deletions in dark blue and some heterozygous deletions as well. One thing that I'd like to also point out is you do see quite a few hotspot missense mutations in some genes such as P53 and some new targets such as FBXW7 and EP300. So there are some implications there that might have to do with some specifics that I'll hopefully get into. The next pathway really involves like cell signaling and receptors that involve RAS signaling or PIK3CA. And so, again, there are some striking differences between HPV positive and HPV negatives, both at the copy number level and the somatic mutation level. Right away, you'll notice that HPV negative proteins, or excuse me, tumors will have amplifications of EGFR at a much higher rate, but also have not much lower, but quite a bit lower numbers of mutations of PIK3CA, but it still has amplifications and some of these hotspot missense mutations. So the HPV positive proteins or tumors also have more PIK3CA, but they also have more PTEN. So PTEN is a negative regulator of PIK3CA, and it seems like, at least in these HPV positive patients, that there's a mutually exclusive relationship between PTEN and PIK3CA. So you can lose one or the other, at least in HPV positive cases. This slide is mostly focusing on immunity, and that's the innate immunity involved in viral clearing and interferon. So you can see at the top, actually there is, it's not entirely split between HPV negative and HPV positive for some of these MHC class 1 genes, such as beta-2 microglobulin and HLA-A and B, but you do see more loss of function and hotspot mutations in the beta-2 microglobulin in the HPV positive cases. So these are probably more likely to impact the function of that gene. But what is quite striking is the locus that involves FAD, BRC3, and BRC2. I think it's chromosome 11q, actually is amplified in a lot of head and neck cancer cases, but these primarily appear to be HPV negative. And you can see on the right that locus is actually deleted in a homozygous fashion. And it's kind of hard to decide what's the real target of this deletion or this amplification, because there are quite a few genes at this locus. But one thing we do see is that there is a striking difference between HPV positive and negative. And along those lines, in the literature, you'll see that BRC2 and 3 are actually associated with promoting a non-canonical NF-kappa B signaling, which can affect type 1 interferon production. So if you're thinking about HPV positive tumors, they'll actually benefit by losing this locus because they can knock out some of the type 1 interferon signaling, and thus the virus never gets cleared, and you end up with cancer. And one of the bits of evidence that also gives us some confidence that losing BRC2 or BRC3 is actually a driver event is you do see some nonsense mutations in BRC3, so the loss of that gene, as well as mutating that gene can have a negative effect on that signaling pathway and promote the virus turning into a tumor. You also see quite a few mutations in FBXW7, which is involved in a lot of different processes. But one of the targets, again, is NF-kB, so there could be some immune effects of this gene. The last pathway I'd like to talk about is the squamous cell differentiation pathway. So One of the most commonly altered genes in squamous cell cancers is Notch1, as you heard earlier. And we do see there are quite a few Notch mutations in the HPV positive cases. However, you see this new gene, ZNF750, which also promotes squamous cell differentiation, getting deleted, as well as getting mutated at quite a high rate, especially in the HPV positive cases. And this relationship, you can see, is quite strikingly mutually exclusive. And so it seems like, at least in these HPV positive cases, for whatever reason, they might lose ZNF750 instead of losing Notch. Again, I've also added some more players to this, but some amplifications that are quite common include TP63 and SOX2, and these seem to be spread evenly between HPV positive and HPV negative cases. So overall, just to conclude a bit, using one of the largest cohorts of head and neck cancer as well as HPV positive head and neck cancer, we were able to identify and expand the list of significantly altered genes in head and neck cancer. Most importantly, we were able to see some recurrently altered targets of the HPV proteins specifically. This involves RBL1 and 2, like I mentioned, as well as some other epigenetic and regulating genes such as EP300 and CRBP, and the FAD caspase-8 signaling pathway. So it seems like in these HPV positive tumors, there actually is a little bit of pressure to mutate some of these proteins that are actually interacting with HPV proteins. So one thing we're trying to incorporate is the viral data that we get. So not only does our sequencing panel allow us to target cancer genes, but we can actually sequence the entire virus sequence. And this information could be useful to maybe correlate some of the properties of the virus, such as viral load, or the intact segments of the viral genome with some of these alterations we're seeing. So maybe samples that have deletions in certain regulatory regions of the HPV genome could have a little bit more pressure to alter these targets rather than depending on the viral proteins themselves. So just some acknowledgements, and thank you for being here.
Hello everyone, my name is Laura Torrens. I'm a postdoctoral researcher at the International Agency for Research on Cancer, and I'm going to present our study entitled Mutographs Head and Neck Project, Understanding the Causes of Head and Neck Cancer Using Mutational Signature Analysis. The known causes of head and neck cancer include smoking and alcohol, as well as human papillomavirus infection, which is predominantly found in oropharynx cases. Epidemiological studies suggest that alcohol and tobacco account for around 72% of the disease burden, with about 33% of this being due to tobacco alone and 35% being due to the combined effect of alcohol and tobacco. Alcohol on its own was found to be responsible for only about 4% of the disease burden, suggesting that alcohol consumption among never smokers has only a limited impact on the head and neck cancer burden. This raises the question of how alcohol exerts its carcinogenic effect and how it augments the known effect of tobacco or human papillomavirus infection. In addition, there is a subset of head and neck cancer cases, predominantly in young women, without a history of exposure to the main risk factors, for which etiology is unknown. Other potential factors, including poor oral health and hot beverages, could play a role in these cases. On the other hand, mutational signatures are a useful tool to track exposure to known and suspected cancer risk factors, including both endogenous and exogenous mutagenic agents. They consist of nucleotide substitution patterns on the cancer genome generated due to mutational processes that have been operative over the lifetime of a patient. In fact, many cancer risk factors have been reported to generate specific mutational signatures, such as tobacco, alcohol, and DNA repair deficiencies, among others. Several types of mutational signatures have been identified in cancer, including single-base substitution signatures, double-base substitution signatures, and insertion-deletion signatures, also known as indel signatures. By figuring out what causes these mutational signatures, we could identify new and potentially preventable causes of cancer. So considering this, here we aim to track the exposure to known and putative risk factors for head and neck cancer across multiple geographical regions and subsites, to evaluate whether an alcohol signature is present in head and neck cancer cases, and to assess its interaction with tobacco smoking or human papillomavirus infection. To do so, we selected a total of 619 head and neck cancer cases from 2 retrospective head and neck studies in Europe and South America, named ARCAGE and Interchange respectively. After a stringent pathology review and sequencing quality control, whole genome sequencing results were generated for 265 cases with available lifestyle and environmental exposure data. De novo signatures were extracted with Seq Profiler extractor and attributed to each sample using MSA. We also used the genomic data to assess the presence of germline alterations linked with head and neck cancer. Finally, we performed multivariate regression analysis to identify associations between the mutational signatures present in each case with epidemiological and germline data. Moving to the results of the study, we identified a high frequency of APOVEC-related single-base substitution mutational signatures, which are depicted here in yellow. These signatures were present in around 90% of head and neck cancer cases in our dataset. They were similarly present in all subsets, which included hypopharynx, larynx, oral cavity, and oropharynx cancer cases. Overall, APOVEC signatures accounted for around 21% of the total mutational burden found in all head and neck cancer cases, indicating that APOVEC-related signaling is a strong mutagenic agent in head and neck cancer. Next, we looked for mutational signatures associated with known head and neck cancer risk factors. In this regard, several mutational signatures were associated with tobacco consumption, including the single-base substitution signature SBS288A, present in more than 60% of ex-smokers and current smokers, compared with 10% of never smokers. Similarly, the double-base substitution signature DBS78A and the indel signature ID83A were also enriched in ever smokers. All these signatures show the mutational profile which has been previously linked to tobacco consumption. Regarding the presence of alcohol-related signatures, a single-base substitution signature named SBS288I was enriched in never drinkers when compared with never drinkers. This was also the case of the double-base substitution signature DBS78C and of the indel signature ID83E. In all cases, the signatures were present in both ex-drinkers and current drinkers. Again, the substitution profile of these signatures has been associated with alcohol-related mutagenesis in other cancer types. Notably, the burdens of SBS288I and DBS78C were higher in head and neck cancer cases from patients exposed to both tobacco and alcohol compared to those exposed to alcohol alone. In this sense, 60% of ever smoker, ever drinkers presented the signatures compared to 25% of the cases from ever drinkers that did not smoke and 0% of the cases of patients not exposed to alcohol or tobacco. A rare germline variant in ADH1B encoding for an alcohol dehydrogenase was identified in 10% of the European cases and 5% of the South American cases. This variant has been previously linked to decreased head and neck cancer risk due to fast alcohol metabolism. We observed that head and neck cancer cases with this variant presented low SBS288I burden as well as low DBS78C burden. In fact, only one case presenting this rare variant was positive for the single-base signature and 4 cases for the double-base substitution signature. This ADH1B variant was also significantly associated with a decreased alcohol consumption measured as grams per week. This is in line with previous studies reporting a protective role of this variant for alcohol-related head and neck cancer. Next, we assess the presence of specific mutational signatures associated with potential head and neck cancer risk factors. For instance, we identified 3 signatures with a mutational profile characteristic of UV light exposure, which were the single-base substitution signature SBS288C, the double-base substitution signature DBS78B, and the indel signature ID83F. All 3 signatures presented a particularly high burden in oral cavity cases. In particular, most cases were located in the internal lip, in the floor of the mouth, and in the tongue, and only one case was identified from the external lip. The presence of these signatures was also associated with high mutational burden, a trait which is also common in UV-related cancers. We also assessed whether there was an association between poor oral health and mutational signatures. To do so, we calculated an oral health score based on the use of denture, gum bleeding, annual dentist visits, and daily toothbrushing. We observed that cases with low oral health score presented high burden of SPS288E, which is a potentially novel single-base substitution signature. In fact, 50% of samples with low oral health score were positive for this signature. In conclusion, mutational signature analysis points towards ApoVex signaling, tobacco, and alcohol as strong mutagenic agents in head and neck cancer. Our study provides molecular evidence suggesting that tobacco could enhance the oncogenic effects of alcohol, and that poor oral health as well as UV exposure may act as putative risk factors for head and neck cancer. These findings have the potential to help unravel preventable causes of head and neck cancer and mutational signatures related to its exposure, and to improve our understanding of mechanisms involved in the carcinogenesis of these tumors. And finally, I would like to thank all the team members and collaborators involved in the study, including colleagues from IARC, Sanger Institute, UCSD, and the Interchange and ArcAge participating centers. Thank you very much for your attention.